Hi guys. A very common question in biology is how does a long DNA get accommodated into a small microscopic nucleus? It's really strange and it is surprising that a DNA, say suppose if we consider a human DNA, it measures around 2 billion nanometers in length and it gets accommodated within a small microscopic nucleus which measures around 2000 to 6000 nanometers in diameter. Now, in order to get condensed and get accommodated within the microscopic nucleus, it um, follows various stages of condensation. Now, in this session, I'll be just discussing about what are the stages through which it passes so that it can uh, get accommodated within the small microscopic nucleus. To understand the various stages of condensation, the first thing we have to know is the definition of the term packing ratio. Now, what is this packing ratio? Say, suppose we have a initial DNA measuring, say, suppose uh, x nanometers in length. Sorry, x nanometers in length. Now, it passes through various stages of condensation. It passes through various stages of condensation, and during mitosis it gets condensed into a very small compact form during which it is called as a chromatid. And during the stage of chromatid, when it exists in the form of a chromatid, it is very small and condensed. So from this big length it gets condensed into a small form so that it can get easily accommodated within the nucleus. So the packing ratio is nothing but the ratio between the initial length of the DNA and the final length, which is obtained after the process of condensation. To understand it better, let us take a human chromosome, shortest human chromosome, and it has been shortest human chromosome containing a number of base pairs within it, and it has been found that this shortest human chromosome contains the base pairs. The number of base pairs within this shortest human chromosome come to 4.6 into 10 to the power of 7, and the distance between any two base pairs comes to 0.34 nanometers. So in order to obtain the distance of the complete or the length of the complete DNA, we have to multiply this 0.34 nanometers with the number of base pairs. Here in this case, it is 4.6 into 10 to the power of 7. Number of base pairs is 4.6 into 10 to the power 7. So it comes to around 14,000 micrometers, the length of the DNA. 14,000 micrometers in length. So the length of the shortest DNA has been found to be 14,000 micrometers. Now it gets condensed ultimately during mitosis. It has been found that it gets condensed to a length of 2 micrometers. So we can see from 14,000 micrometers, it gets condensed to 2 micrometers. So the packing ratio over here is 14,000 divided by 2, which comes to, so the packing ratio is 7,000. So it gets intensely packed, the human chromosome gets intensely packed to get accommodated within the microscopic nucleus. So to understand that, let me move to the next slide. So in this slide I had tried to give the definition of the packing ratio. The ratio obtained when the length of the DNA is divided by the length into which it is packaged. And as an example, I have explained about the shortest human chromosome. There are 4.6 into 10 to the power 7 base pairs present in the shortest human chromosome. To obtain the length of the DNA, the number of base pairs is multiplied with the 0.34 nanometers, which is the distance between two base pairs. And hence the total length of the DNA comes to 14,000 micrometers, and the most condensed form of DNA is obtained during mitosis, and it measures 2 micrometers in length. Hence the packing ratio comes to 7,000. Now moving to the next slide. Now, in order to come to such a condensed form, it undergoes through various steps of organization. So, the organization of DNA, and this kind of organization is different in the case of prokaryotes and is different in the case of eukaryotes. Prokaryotes are nothing but single-celled organisms which do not have a well-defined nucleus. So DNA is organized into a structure called nucleoid. As they do not have a well-defined nucleus, the DNA gets organized within the structure called nucleoid. The negative charge of DNA is counteracted by the positively charged polyamines such as spermine and spermidine. So in the case of prokaryotes, the DNA is basically both in prokaryotes and eukaryotes, DNA consists of sugar phosphate backbone, that is a single chain of sugar phosphate backbone, and the various nitrogenous bases which project outside almost perpendicularly. So it appears like this, that is, we can say that a sugar is linked to a phosphate, and then a sugar phosphate backbone, they are linked together, and the bases protrude out perpendicularly. Various bases, adenine, guanine, cytosine, thymine, these bases protrude out from these sugars, from this sugar phosphate chain. So, and this phosphate group is responsible for the negative charge of the DNA. The phosphate group is responsible for the negative charge of the DNA. Negative charge of the DNA, and this negative charge is counteracted by the positively charged polyamines such as spermine and spermidine. Now, abundant small proteins like HNS give a compact supercoiled structure to the DNA. So, apart from these polyamines and spermine and spermidine, there are abundant small proteins also, like for example HNS, which ultimately give a compact supercoiled structure to the DNA. And DNA gyrase is an enzyme which opens up the DNA during replication. We know that DNA is arranged in the form of a double helical structure. So DNA has a double helical structure, and it is quite intricately compact. That is, in the case of prokaryotes, the polyamines and the small proteins like HNS all try to give a very compact structure to it, and the DNA gyrase is the enzyme which opens up and separates the DNA. So what is the enzyme? That is DNA gyrase is an enzyme which opens up and makes it ready for the replication. Opens up the DNA during replication. Now moving to the next slide. This is a slide which shows the structure of polyamines which interact with the DNA. As I said, the DNA has a negative charge because of the phosphate groups. Now the negative charge is neutralized with the help of polyamines like spermine and spermidine. We can see that both of them are abundant. They are basic in nature and they have positive charges, as a result of which they neutralize the negative charge of DNA. This is the structure of spermine and spermidine. Now moving to the next slide. Organization of DNA within eukaryotes. Now this was the discussion about organization of DNA in case of prokaryotes, that is those organisms which do not have a well defined nucleus, whereas now coming to eukaryotes which have a well defined nucleus, the DNA is present along with some basic proteins in the form of chromosomes within the nucleus. So within the nucleus, well defined nucleus, the DNA is associated with some of the histone proteins, and this coming to chromatin, it is a unit of analysis of chromosome and gives a general idea about the nature of the chromosome. So the chromatin is nothing but a single unit of analysis. That is, say suppose if we consider a chromosome, this is a chromosome during the mitosis stage, wherein the DNA has got replicated and sister chromatids have got originated. So the chromatin is nothing but a small unit of analysis within the chromosome, and the chromosome is entirely the DNA along with the histone proteins gets compactly arranged within the nucleus. So moving to the next slide. Histones. What are these histones? They are positively charged basic proteins. Now, as I said, the DNA is negatively charged because of the phosphate groups. Now, in the case of eukaryotes, this negative charge is neutralized with the help of positive charge of the basic proteins, which are histones. Now, histones are basically of 5 major types, and they contain amino acid residues like lysine and arginine. These are basic amino acid residues, lysine and arginine. And histones are the 5 major types of histones are H1, H1, sorry, H2A, H2B, H3, and H4. Now, formation of nucleosome. This formation of nucleosome is the very first step of organization, or you can say the first level of organization of the DNA. So, the histone proteins form an octamer, that is 8 molecules of histone, 4 towards the front end and 4 towards the back end. So completely they form an octamer consisting of 8 histone molecules, and the DNA gets wound around the histone proteins. It takes a one full turn, and then it covers the 3/4 of the octamer, and then again wraps around the next nucleosome. So this is what is called as nucleosome, which consists of histone octamer and the DNA wrapped around it. So and this is the fundamental unit of chromatin. That is, chromatin is made up of number of nucleosomes. So histone octamer acts like a core and consists of 2 copies of each of these histone proteins. That is, it contains 2 copies of H2A, 2B, 3 and 4, finally forming 4 histone molecules, and around which the DNA duplex wraps around it. So the duplex DNA which is wrapped around the histone octamer is approximately 147 base pairs in length. This is a small basic unit and is termed as a nucleosome. Nucleosomes together, So the nucleosomes together with DNA appear like beads on the string. At this stage, the DNA is 10 nanometers in diameter and attains a packing ratio of 6. So here we can see that DNA was initially a single chain, in a single long chain. Now this has attained at an organization level wherein it measures around 10 nanometers in diameter and attains a packing ratio of 6. And here we can see very much it appears like a necklace, isn't it? Wherein a small number of beads, large number of beads are there, and these beads are joined together with the help of DNA. So these beads are nothing but nucleosomes, and the DNA helps to join the nucleosomes. So this is a stage of first level of organization, that is the formation of nucleosomes. Now moving to the next level. Here I have just tried to show the structure of a single nucleosome. I'll just zoom it. So this is the DNA, double helix DNA, which wraps around the histone octamer, the histone molecules, and we can see that DNA has taken one complete turn. It starts from here, it has taken one complete turn, and in the next round it covers the 3/4 of the octamer, and then it protrudes for the next nucleosome. And this is histone 1, which is a linker histone. I'll be discussing about it later. So this is a structure of the nucleosome. Now moving to the next level of organization, that is, now at this stage I'll be discussing about the linker histone or histone 1. It holds the DNA which is wrapped around the nucleosome in the proper position. So when the DNA wraps around the nucleosome, it has to be held in the proper position, and that is done by histone 1. The linker DNA is made up of approximately 20 to 60 base pairs, and the H1 binds to the linker DNA. So this DNA which exists between the nucleosomes, this is nothing but the linker DNA, which is linking the nucleosome to another nucleosome. And this linker DNA is small, which measures around 20 to 60 base pairs in length, and H1 binds to the linker DNA. So as it was shown in this figure, this gives stability to the zigzag 30 nanometers chromatin fiber. So the next stage of it is the 30 nanometers chromatin fiber, and to give the stability to it, this linker DNA helps in giving the stability to the 30 nanometer chromatin fiber. So this is the linker DNA which links one nucleosome with the another one, which measures 20 to 30 base pairs in length, and histone 1 binds to this linker DNA, not the actual DNA which wraps around the histone octamer. And this gives a sort of a stability to the next organization level, that is a solenoid fiber or 30 nanometer chromatin fiber. So formation of the chromatin fiber or the solenoid fiber, this is the next stage. As I said, from the beads on the string stage, the DNA coils in such a way that at least 6 nucleosomes are packaged per coil. So what happens is, at this stage, this is a beads on a string stage. Now slowly the DNA starts like this. 6 nucleosomes per coil get coiled. So in the next stage, the DNA coils in such a way that at least 6 nucleosomes are packed per coil. So per coil we have 6 nucleosomes. In this stage, the DNA attains a diameter of 30 nanometers, and this level of organization is known as 30 nanometer fiber or solenoid fiber. When the chromatin is extracted from isotonic buffers, it appears it appears like a 30 nanometer fiber. So when it is extracted from the isotonic buffers, it appears like a 30 nanometer fiber. At this stage, it attains a packing ratio of 10. So here we can see that the packing ratio has increased from 6 to 10. This stage is seen during interphase stage of the cell cycle. So during the interphase stage, that is during the preparatory stage of mitosis, before the actual cell division, the cell prepares itself. During this preparatory stage, the chromosome takes in this 30 nanometer fiber stage. So here at this stage, the diameter here at this stage, that is beads on a string stage, it was 10 nanometers in diameter. Now at this stage, the diameter comes to 30 nanometers. And the packing ratio comes to 10. So from the 6, the ratio of packing ratio to, from the 6 to it has moved to 10. Now, what are the stages after the solenoid fiber? Now, after the solenoid fiber, it gets further coiled and condensed and is organized into loops, scaffolds, and domains to obtain cytologically visible threads known as chromatids. So, during mitosis, the DNA obtains a structure known as chromatids, which are easily visible under the microscope, under the light microscope. And to obtain such stage, the solenoid fiber gets further condensed and coiled. So and this the looping is such that the base of the loop is attached to the same protein skeletal work. Some metallic ions like calcium and copper along with non-histone proteins help in the looping process. So such sort of loops, just to explain this, like this, these loops are arranged. This is the next stage wherein each of this loop is having 6 nucleosomes. These loops around the nuclear matrix. Here we have the nuclear matrix. Around this nuclear matrix, it has got looped in, and the very next stage of it is it forms a mini band. Like say suppose we have in this center we have the nuclear matrix. Say suppose this is the core of the nuclear matrix, and then we have number of loops. So such will be the appearance of a mini band, and many mini bands together form a compact chromosome. So suppose this is a centromere, and these are the chromatids. Now these mini bands occupy occupy many millions of mini bands are present within these chromatids, right? So this is how the organization, this is how the various stages of organization, through various stages of organization, the DNA attains a very compact form. So coming to the PPT, so it gets further coiled and condensed and is organized into loops, scaffolds, and domains to obtain cytologically visible threads known as chromatids. The looping is such that the base of the loops are attached to the same protein skeletal work. Some metallic ions like calcium and copper along with some non-histone proteins help in the looping process. This increases the packing ratio to about 1000 in the interphase stage, that is during the preparatory stage, and to 10,000 in the mitotic chromosomes. So in the preparatory stage, the DNA is still loosely packed, whereas when it comes to mitosis stage, it gets very compactly packed, and at this stage we can see that the packing ratio comes to 10,000. So you can imagine to what extent it gets packaged to get accommodated. Now moving to the next slide, this is a diagrammatic. This in the slide it will show diagrammatically the various levels of organization. I'll just focus it so that we can view each of the stage. So this is a double helical stage, initial stage. This is 2 nanometers in diameter, and it is 2 nanometers in diameter, and we have 10 base pairs per turn, and the packing ratio is 1. Now moving to the next stage, that is beads on the string stage, where we can see that the DNA has wrapped around the nucleosomes, and it appears almost like a chain containing number of beads. And here we can see that the base pairs per turn are 80, and the packing ratio is 6 to 7. And the diameter varies from 10 to 11 nanometers. And the next stage is the 30 nanometer solenoid fiber, wherein 6 nucleosomes are accommodated per loop. It coils, and each coil has got 6 nucleosomes per turn, and during this the diameter is 30 nanometers, and here we can see that the number of base pairs per turn has increased. It comes to 12,000, and the packing ratio has also comparatively increased. It comes to 10. So from 6 to 7 the packing ratio has moved to 10. The next stage, it forms loops. So the number of loops are here, and these looping takes place across the nuclear matrix. As I said, that is the nuclear matrix is the base, and around this the coiling takes place. And at this stage the diameter comes to approximately 0.25 micrometers. So we can see that the diameter has increased, and the number of base pairs present per turn have also increased, and the packing ratio has increased to 680. Next comes the mini band. Mini band is at this stage where it has got intricately coiled, and such sort of mini bands are arranged in the chromosome at each step. Millions of such mini bands are present, which ultimately give the structure of chromatid in the case of mitosis. And when it enters into the mitosis stage, we know that it attains a packing ratio of around 10,000. So during the mitosis, it is very compact and intricately condensed. It is present in a very compact and intricately condensed form. So moving to the next slide. So this is this brings an end to my discussion over here, wherein I have discussed about the various stages or various levels of organization through which the DNA undergoes in the case of eukaryotes, so that it can get well accommodated and within the compact nucleus. So the various levels of organization start from beads on the string to and they end up forming a chromatid or a chromosome, a highly compact chromosome.